So good morning, friends. So our today's class is about molecular markers and their application in agriculture. So molecular markers. So in this class we will discuss about what is molecular marker, how molecular markers get developed, and what is their application in agriculture. So why we need to study these molecular markers? We need to understand the concept of molecular marker, how we are using the molecular marker in the crop improvement. So molecular means it may be DNA, RNA molecules, biomolecules, different biomolecules. We are using them as a marker. Marker means we have to mark something or tag something to identify it. Okay, so that is the molecular marker. So molecular markers are nothing but these are the DNA sequences, a specific DNA sequence that is present within the gene or near the gene that act as a tag to that particular gene. And these molecular markers follow the Mendelian pattern of inheritance. They follow the Mendelian pattern of inheritance. So in this regard, we will discuss on different class of molecular markers. What is the marker? So molecular markers are defined as specific DNA sequences that are present within the gene or near the gene, and they follow the Mendelian pattern of inheritance. And the markers are present in a polymorphic form. Polymorphic form means they are in a one individual they are present in a particular sequence, while other persons they have different form or different allele of that gene. So these two different forms act as a markers for that particular gene. So earlier we are using biochemical as a marker. So biochemical markers, morphological markers, and molecular markers. Biochemical markers are different class of marker like isozymes and protein binding pattern. So these biochemicals like protein, enzymes, different form of enzymes, isozymes, proteins, and protein bending pattern on the gel. The how they behave on the gel. Phase means polyacrylamide gels. How the protein, a particular protein, behave on that gel, and then what is their bending profile that is represented in a biochemical markers. So unique bending pattern for a particular trait, for a particular plant, or particular species represent that this protein act as marker, a biochemical marker for that trait. Suppose if we talk about isozymes are nothing but they are different isolate form of the enzymes. Okay, like alcohol dehydrogenase has six different form of the enzymes. Lactate dehydrogenase has six different form of isozymes. So all these forms act as a biochemical marker. Similarly, morphological marker. Morphological as morphology itself indicating morphology. Morphology means plant morphology. Means plant height. What is the plant height? What is the flower color, pod shape, pod size, seed color, seed weight? Okay, so all these morphological traits, plant height, plant morphological appearance, okay, growing habit of the plants. So all these traits act as a morphological marker. So these morphological marker can be visualized by our naked eyes. Okay, so we call them as a naked eye polymorphism because polymorphism is nothing but different form of a particular trait. We have like a polymorph. You can say polymorph. So the morphological traits we can visualize by our eyes. We can easily see by our eyes. So these morphological markers are also called a naked eye polymorphism. Similarly, molecular marker. So later on, when the molecular PCR discovery, molecular techniques has been discovered, after that these molecular techniques or molecular markers is comes into the picture. Okay, so later on this characterization of RNA, DNA, proteins leads to development of these molecular markers. Okay, these molecular markers are generally used recent advanced molecular techniques. Okay, So again, we can classify the institute based on hybridization, based on recent technique, that is PCR, polymerase chain reaction. Based on hybridization means by hybridizing a particular DNA probe DNA sequence to all the length of the DNA based on their sequence complementarity. So this is the basic principle of hybridization of these three molecular markers: RFLP, mini satellite, and microsatellite. RFLP represent restriction fragment length polymorphism. Restriction fragment length polymorphism. Restriction means the restriction fragment of the DNA. Suppose we have a lot of the genomic DNA along with us, then we have to use the restriction digestion for. And what are the different restriction fragments we got, and their polymorphism, their length polymorphism is represented RFLP. Similarly, mini satellite, microsatellite. Microsatellite also known as mini satellite is also known as VNTR, variable number of random repeats. So in the case of mini satellites means long DNA sequences, suppose the GACT. This sequence is repeated GACT 10 to 12 times, 1000 times. So the length of the DNA gets increased because of the repetitive DNA fragments, repetitive sequences. Microsatellite is in this case the microsatellite the repeat unit is small. It may be mononucleotide, dinucleotide, AA, GG, dinucleotide, two nucleotides, set of two nucleotides. It may be GGG, GCT, TC, likewise. Dinucleotide, tetranucleotide. So small repeat unit is there in microsatellite, and that is repeated 10 to 50 times more. So that unit is a complete unit is complete set, or the repetitive unit is known as microsatellite. Okay, so this mini satellite and microsatellite is very important. Mini satellite variable number of tandem repeat is very important in case of DNA profile 7 glottal. Okay, variable number of tandem repeats. Then further, as technological advancement, few more molecular marker has been developed by using the PCR. PCR, everybody is very well aware about the PCR. That is polymerase chain reactions. You know very well. This PCR technique is discovered by the Kelly Mullis, developed by the Kelly Mullis in 1983. Okay, PCR is nothing but it's a thermal cyclone. Thermal cyclone. It's a thermal cyclone. So by using this PCR, lots series of molecular marker has been developed, like SCAR marker, AFLP marker, SSR marker, STS marker, ISR marker, EST marker, RAPD marker, SCOT marker, APPCR. AP means arbitrary primed PCR, SCOT sequence codon, sequence characterizing codon type sequence, RAPD randomly amplified polymorphic DNA sequences, EST express sequence tag, ISR inter-sequence repeat, sequence tag site, simple sequence repeat, SR, AFLP, amplified fragment length polymorphism, sequence scar marker, sequence characterized artificial amplified region. These are the molecular markers developed through the PCR based on PCR. Okay. Further, the molecular marker has been classified based on the molecular cytogenetic technique, using molecular cytogenetic technique and molecular marker techniques. So, using the cytogenetic techniques, fish can extend DNA fiber fish. Fish is fluorescent in situ hybridization. Fluorescent in situ hybridization. Extended DNA fiber fish. Molecular marker techniques: RFLP, RAPD, SSR, AFLP, Scott marker, EST marker. We are using molecular marker techniques. So we are using all these techniques. Fish is nothing but it's a fluorescent in situ hybridization. So in this case, complete chromosome we are using at a time. Complete chromosome we are using. So here, if we have a sequence probe, and that so sequence probe is complementary to hybridize with the based on sequence complementarity. Extended DNA fiber fish means in this first we have to use the interphase chromosome. Interphase means long stretch chromosome, but while in case of fish we are using metaphase chromosome, that is highly condensed chromosome we are using for fish. Okay, so nothing much. This is the best example of morphological markers. As I told you, morphological markers means which is can be visualized by our naked eyes. Okay, these are the morphological markers. You can see here flower color. leaf color, leaf shape, leaf size. Okay. So best is represent the morphological marker. Molecular markers are nothing but these are the exploitation of the genetic diversity. We have to identify, we can harass, we can exploit the genetic diversity by using all these molecular markers. So assessment of genetic diversity in a crop species is fundamental to its improvement. Whatever genetic diversity is present in a particular crop species, that all genetic diversity must be assessed. Genetic variability that is considered to reservoir plant breeders fall upon their continuous strive to develop the improved variety and hybrids. Genetic variability that is present among the different genotypes that we have to use for the crop improvement by through the different crossing hybridization pattern. Knowledge of germplasm diversity and of relationship among the elite breeding material has a significant impact to the improvement of the crop. The germplasm diversity is providing a very good knowledge of relationship among elite breeding materials. This information is useful in planning process for the hybrid and line development and assigning line to heterotic group, assigning the Now we discuss morphological markers. As I told you, these are the related to the shape, size, color, surface of various plant parts. So in case of it may be seed, it may be flower, it may be fruit, it may be leaf, it may be plant growth habit also. Okay. So these morphological markers can be easily seen by our eyes, naked eyes. Such characters used for the varietal identification. These characters used for the varietal identification. Advantages: they are readily available. Morphological markers are readily available. Usually require only simple equipments. Form the most direct measure of phenotype. Readily available. Usually require only simple equipments. There is not much complex equipment. Form the most direct measure of phenotype. Disadvantages: it require expertise on the crop or species. Subject to environmental influence, limited in the number, limited genomic coverage. Protein markers, or in other words, we can say it's a biochemical markers. Okay. These biochemical markers are available since 1950s. Such markers are related to the variation in the protein and amino acid. These markers are related to the variation in protein and amino acid binding protein. Gel electrophoretic studies used for the identification of biochemical markers. Gel electrophoretic studies used for the identification of biochemical markers like peroxidase, ester phosphatase, esterase. All these act as a protein molecular markers. So the gel electrophoretic studies can be easily used among the for identification of biochemical markers like peroxidase, ester phosphatase, esterase. So these protein act as a protein biochemical markers. So advantage, if we talk about the advantage of these protein markers, first advantage is requires simple equipments. There is not much complex, simple equipments. Biggest complement to the morphological assessment of the variations. Biggest complement to the morphological assessment. Disadvantage is subject to environmental influence, limited in the molecular. Now molecular markers. Molecular markers reflect the heritable differences in homologous DNA sequence among the individuals. They reflect heritable differences. As I told you, molecular marker are nothing but they are the sequence difference which are heritable according to Mendelian inheritance pattern homologous DNA sequence among the individuals. They may be due to base pair changes. Base pair changes. So these differences, sequence differences, is because of base pair changes, because of rearrangement, translocation, or inversion. Rearrangement of the DNA sequences. The rearrangement is because of either translocation or inversion. Then insertion or deletion. Insertion of a single nucleotide or deletion of a single nucleotide leads to development of insertion deletions. Then variation in the number of tandem repeat. Variation in the tandem repeat. Variation in tandem. If we talk about advantage of molecular markers, they are ubiquitously present. Ubiquitously means they are present throughout the genome. Means ample amount. How much we can explore all the molecular markers. So all these molecular markers are nowadays we are identifying a lot of molecular markers because of the sequencing data. Because we have lot of the sequencing data. As a particular crop got sequenced, we can identify particular sequence, use them as a molecular markers. So as the sequencing data is available along with us, so we can develop a lot of molecular markers. So they are ubiquitously present throughout the genome. Stably inherited. Stably inherited means because they are present in the genetic material, because they belong to the genes, sequence, coding sequence, coding sequence, or non-coding sequence. So these are the ultimate part of the genetic material. So they are stably inherited into the next progeny. Okay. And they follow Mendelian pattern of inheritance also. Multiple allele for each markers. So here, as I told you, genes have different alleles, means different form of a genes is known as allele. So here, a particular genes can have more than one allele, more than two allele, polymorphic gene also is there. So multiple allele for each marker is also available. Maximum, we have single nucleotide can get replaced by three markers. We have three or four. Okay. So similarly, if a particular nucleotide gets changed, instead of that, we have pyrase, purines, or pyramids. Multiple alleles can be generated for a particular markers. Devoid of clear trophic effect. It will do not have clear trophic effect. Detectable in all tissue at all stages because as they belongs to genetic material present, same genetic material present in all the tissues, all the somatic cells, gametic cells, at all stages of the plant. That is different thing that a particular stage that is expressing or not. That is a different issue. But they are present every time they are present in each and every tissue at all stages, all growing stage of the plants. So that is the one most advantages of this molecular markers. Okay, they are present throughout the genome. They present in all the tissue at all stages of the growth. The long shelf life of the DNA sample. As suppose we have isolated the DNA, we have kept for the for the future use, so we can keep. And the DNA samples cannot be easily degraded for a longer time. While the RNA sample is easily degraded, we cannot store for longer time RNA. DNA can be stored for longer time. These are the few advantages of molecular markers. So we can use them. They are stably ubiquitously multiple for each markers, and they do not have environmental influence. Exactly, the much environmental influence is not there. But environment or genetic interaction is there, but direct influence of environment is not so much in that molecular markers. So that is another advantage of it. Now, if we talk about the conventional molecular breeding here, so we can make a compare. So to understand better understanding of the molecular markers, because we are using them as in a molecular breeding. Conventional breeding is a different from the molecular breeding. In the case of conventional breeding, generally we are using morphological markers more commonly. But in case of molecular breeding, we are using molecular or DNA markers more as if we are using morphological means not a simple laboratory is required. While in case of molecular markers, we have to use sophisticated instruments, sophisticated laboratory there. is needed. If we talk about effect of environment, as I told you right now, the molecular markers do not influence much by the environment. While the morphological marker, morphological data, definitely they have high effect of environment on the conventional marker. There is no effect of environment on the markers. Accuracy of method, we talk about the accuracy of method, medium to high, very high. Time required to develop new varieties. It's require very conventional breeding require very long time, while molecular breeding require very short time, 3 to 5 years. Because it reduces the so many of breeding cycles of the particular crop. Cost involved low to medium as high sophisticated instrument required in case of molecular breeding, so cost is going to be very high. Health agent only mutational breeding. The technique involved radioactive labeling like that. The effect of gene interactions is very high in case of conventional, while in case of molecular breeding, no effect. Mapping of fetal is not possible. In this case, possible by using the molecular markers. Screening of seedling stage for economic trait. Yes.